Good morning or afternoon. Okay, are there any questions? Well, let's give students a little longer to join. For those of you who just joined, are there any questions? Am I loud enough?
Yes.
Okay, thank you. I'll turn it up a little. Let's see. Let's first talk about what we're going to be covering. Today is week 3. We'll be covering chapter 6. This week you have quiz 2, which is due on Friday. It will covers chapters 4, 5, and 6. Later in the week, we'll be covering chapter 8. That will not be on quiz 2. And quiz 2 also covers lab 2 and lab 3, which were the labs you did last week. Any questions about that or what quiz 2 covers? Let's put in something right here.
Also have a practice quiz up, correct, on the 17th?
Yes, I'm going to put that in here.
Is that worth like extra credit, or is it just for us to like practice and get ready for the test?
It's both. So let me put that in here. Of course, I'll have to update your practice quiz 2. And I don't remember when that's due. Does anyone remember?
17th, I believe.
17th? It's right here. Yeah, 17th. That one is open. Oops, wrong one. So all right, that got that. So that's in there. Let's see if I can make that a little neater. Nope. So let me talk a little bit about the practice quiz. First, are there any questions about quiz 2 or what we're covering today? This is Monday, so we're going to be covering lab 4 in the second part of the class. And that'll be on stains. And hopefully you read textbook chapter 3 through the end of the section on the part about microscopes. All right, if there's no questions about quiz 2 or what we're covering, let me talk a little bit about practice quiz 2. And I've got that up here. So you can find it or the link to it under week 3 of the lecture. And it's only 5 questions. Oh, it says 0 points, but I'll give you up to 2 extra credit points if you take the quiz. Let me open it here. I think it's already open, meaning you can take this one at any time. Oh, that's showing the answers. Available from June 15th. So yeah, it opened at midnight this morning and will be up until June 18th, 11:59 p.m.
What is that lockdown browser it says it requires?
Yes. So it's going to be testing the lockdown browser as well as giving you a practice for quiz 2. And It is a time test, but there's only 5 questions and you're getting 9 minutes. So that's much more generous than your other time tests. It will be a practice for quiz 2. It's covering the same areas. One of the questions will be the same question that's on quiz 2. So there's 2 reasons to take it. First, you'll get some practice points. And second, like I said, it really will be a practice for quiz 2. All right, any other questions about it? So you find the link to it here. Please take that and let me know how it goes. And then quiz 2B, you're seeing something that's not published, so let me get that out of the screen. Quiz 2B is the real test, and it'll be 25 points, 25 questions. It'll be worth 50 points. I haven't finished the quiz, so that's why it's saying 30 points. And it is due by 11:59 p.m. on June 19th. Now, if you take the practice quiz, you'll be set up to take quiz 2B. If you don't take the practice quiz 2, when you are getting the Respondus Lockdown browser, there is a download that you have to take these 2 quizzes. That will lock down your browser, and besides the quiz running, you will also have the lockdown browser running. So you shouldn't have a whole lot of things running on your computer. I don't think this will run on a phone. I don't think you would take a quiz on a phone, but I don't think this will run on a phone. But I have set it up that supposedly, what is it called? A Macintosh or an Apple iPad. It should work on an Apple iPad. I don't know about other iPads, but it will work on the Apple iPad. Any questions about any of that? All right. So those are some things coming up, and that tells you what we're doing. Let's go ahead and get started with chapter 6. Are there any questions on anything we've dealt with previously? All right. Let's do that over there. So chapter 6 is going to cover microbial growth. This slide is showing you the major goals of this chapter and a rough outline of how we'll be covering it. Know the terms psychrophiles, psychotroph, mesophiles, thermophiles, hypothermophiles, acetophiles, halophiles, what an aerotolerant anaerobe is, a facultative anaerobe, and other oxygen requirements for growth. Binary fission, reproductive spores, and endospores. We've already talked a little bit about endospores, but we'll talk about it in greater depth today. Understand the various requirements of cells for growing, understand the 4 phases of growth, and compare the different methods of measuring microbial growth. Is there any question about any of that? Get a little more light in here. All right. When we're talking about microbial growth, you should realize when we're talking about microbiology, when we mean microbial growth, we mean an increase in the number of cells, not the cell size. A long time ago, I used to teach middle school students, and when we talked about them growing, we didn't talk about them reproducing. Okay, any questions about that? Let's take a look at a slide. I think I should probably get this going here, and then it's a little easier. time-lapse photographs of bacterial growth. Assuming that it loads. I don't know why it takes so long for that to load. Did anybody hear that?
No.
All right, that means I don't have the sound on, right? Let me go back to this. Come on. Oh, it's right here, sorry. Stop the share. I think I start the share. I'm in my class. Yes, sorry about this. Can you enter the door? Because I'm in my class. All right. I'm sorry?
Is the freezer, Charles freezer is up?
No, I'm the top shelf and part of the door. Sorry about that, glass. Something going on with my home. And how do I start this again? That doesn't look good. Can you see the screen?
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
All right, any questions about that? I assume this time you could hear it. When we're talking about microbial growth, all things when they're growing have certain requirements, and there are physical requirements. That includes temperature, pH, acidity, well, I guess that's pH, alkalinity, and salinity. So those are physical elements that things need to grow. There are also chemical elements that things need to grow. Chemical sources, like all microbial growth, they have to have a carbon source if they're to grow. They have to have a nitrogen, a sulfur, a phosphorus source, a source of trace elements. And if oxygen is required by the microbial growth, they have to have a source of oxygen. And there may be organic growth factors that are required. Like for humans, we have to have vitamin C. That would be an organic growth factor required for our growth. Any question about any of that? If you were in a microbiology lab and growing things in the lab, the media would supply all of these physical and chemical sources that are required by the cells to grow. Most of the cells you would have grown in the lab would not have had an organic growth factor required. If we were to grow something like Neisseria gonorrhea, that would have organic growth factors that are required. But E. coli can survive with the just like the beef extracts that are included in the media. Any question about any of that? There are physical requirements. Temperature is a physical requirement of things to live. And each species of bacteria has its minimum temperature it will grow at, the maximum temperature it will grow at, and an optimum temperature that it will grow at. The optimum temperature is when the species will grow the best. The minimum temperature will be the lowest temperature at which the species would grow. Below that temperature, the species would not grow. It might not die, but it would not grow. You can classify microorganisms into groups on the basis of their preferred temperature range. Any question about that? So psychrophiles, let's see if I can blow that up just a little. Psychrophiles are organisms that grow at very cool temperature. These would be microorganisms that grow in your refrigerator, and they can cause food spoilage. The range of the different temperature requirements is not hard, fast, but generally speaking, psychrophiles are things that grow at 10 degrees Celsius or less. And like I said, that's refrigerator temperatures. Generally speaking, below 0 degrees, things don't grow unless they have an antifreeze in them, because 0 degrees is when water freezes. But there are things that can grow about minus 10 degrees Celsius because of antifreeze. And like I said, the psychrophiles grow, well, the optimum is between 4 and 10 degrees Celsius, and they can grow up to about 20 degrees Celsius. The psychotrophs are things that grow a little bit warmer temperature, and their optimum growth temperature is at about 20 degrees. So those would be things more or less living at room temperature. The mesophiles are things that have an optimum growth temperature at 37 degrees Celsius. And what do we know that's 37 degrees Celsius?
Human
Come
body.
on, somebody's got to know. What's 37 degrees Celsius?
The human body.
Yeah, the human body. Inside the human body is 37 degrees Celsius. That's 98.6 degrees Fahrenheit. And all of the human pathogens are mesophiles. Now, you'll notice that the optimum temperature is 37 for your average mesophile, but the organisms that grow on your skin are closer to 30 degrees Celsius, and that would be their optimum, meaning the optimum isn't in stone either. Like the optimum mesophile that grows on your skin would grow best at 30 degrees. So this would be where the highest part of the peak would be for a mesophile growing on your skin. Any question about that? What I'm trying to say is exactly where this peak is will shift a little bit. It's not in stone. For the average mesophile, though, the average would be 37 degrees for the average mesophile. Any question about that? Thermophiles are organisms that grow at very warm temperatures between 60 and 65 degrees Celsius. That would be about the temperature of the chicken when you go to Safeway or some other store and it's cooked and it's on the rotisserie there. That would be the temperature of heat lamps in a restaurant. What should I say about that? Anyways, there are bacteria that can grow at that temperature, and they grow at elevated temperatures. These organisms could not grow in your body because their enzymes and their cell membranes would literally freeze, okay? That it wouldn't be the correct temperature, and so they wouldn't work at your body temperature. The same thing with the psychrophiles. They would not grow in your body because, well, their enzymes and their cell membranes would probably fall apart at 37 degrees Celsius. The enzyme certainly wouldn't work for a psychrophile. The hypothermophiles were something that weren't known when I took microbiology. Tells you how long ago that was. But these are organisms that have a optimal growth temperature at 95 degrees Celsius or higher. And so some of them grow at the temperature that water boils at, okay? So they're really elevated temperatures. Many, if not most of the hypothermophiles are archaea. And of course, these organisms would not grow in us either because their enzymes as well as their membranes would freeze in us. All right, any question about that? When we're talking about temperature, I should briefly talk about the temperature that food spoilage can happen. This can happen. Let me blow this up so it's a little more visible. Food spoilage happens rapidly between about 15 degrees Celsius, maybe 17 degrees Celsius to 51 degrees Celsius. And that would be 60 degrees Fahrenheit to 130 degrees Fahrenheit, where bacteria will grow rapidly. And so when you have food at that temperature, you shouldn't have it out for very long. Food will spoil at other temperatures, particularly between, well, let's say 40 degrees Fahrenheit to 145 degrees Fahrenheit. But at above 130 to 140, bacteria can grow in it and spoil, but the bacteria don't grow very rapidly, so that food takes longer to spoil. And in the refrigerator, as you know, between 0 degrees and what is that? 0 degrees Celsius to, I don't know, 5 or 7 degrees Celsius. That would be 33 degrees Celsius to 40 degrees Celsius. Food can spoil, but it will take about a week for the food to spoil. And that's assuming there's nothing in it to help preserve it, like salt or vinegar or acid. So pickles and ketchup, which both have vinegar and salt in it, they won't spoil anywhere near a week. Any question about any of that? So the idea is that when you have food that you make, you put it in the refrigerator or the freezer or an elevated temperature, although nobody at home has that. And that will slow down the bacteria from growing. In the refrigerator, bacteria will grow, but much slower than at room temperature or at 37 degrees or warmer. And that's why food can stay in the refrigerator for a week without preservatives. Any question about any of that? It's the psychotrophs that generally cause food spoilage in the refrigerator if you don't ever take it out and let it warm up. Mesophiles that are in the food, if you take the food out, they will grow when the food is warming up. And they will also grow when the food is cooling down when it's in the refrigerator. So it's mostly the psychotrophs and the mesophiles that cause food spoilage. I'm gonna skip that. Let me think about this. Let me see if this is where we're starting our, yeah, this is when we're starting the worksheet, so I'm not gonna talk about this slide. Any questions about that and food spoilage? In some of the physical requirements for cells to grow, I've got it in the worksheet. It's pretty straightforward, so I'm not going to talk about those, like acetophiles, things that grow in acid environments. All right, so that's where the end of the worksheet is. So let's move on to the next slide. Chemical requirements. All living things have an oxygen requirement. For an anaerobe, the oxygen requirement is that no oxygen be present. So microbes that use oxygen, we call them aerobes. That literally means they breathe air. And when we mean that, we mean they're breathing the oxygen in the air. And aerobes produce more ATP from the nutrients than anaerobes that don't use oxygen. And so aerobes tend to grow faster than anaerobes. Any question about that? We classify microorganisms on the basis of the effect that oxygen has on their growth. Is that clear? So let me blow this up because it's a little small. This is a thioglycolate tube, and that's a media, a liquid media that pulls oxygen out of the media. That's all you really need to know about thioglycolate. But once you have the tube out of the autoclave, there's oxygen in the air, and it comes into the media. And that means that the top layer of the thioglycolate will be at full oxygen, around 20%. And as you go down in the tube, the oxygen will decrease. And at the bottom of the tube, the oxygen level will be about 0%. So a thioglycolate tube creates an oxygen gradient from 0% in the bottom to around 20% at the top. Any question about that? And then when you put the bacteria in the tube, you stab it with a loop because it's a liquid media. So you put the loop in. I'm trying to get my mouse to work. So you get the bacteria on a loop, and then you put it in the tube, and then you bring it out, and then you cap the tube, and then you incubate it, and then you see where the microorganisms are growing. If they're only growing at the top of the tube, that's an obligate aerobe, meaning that that organism only grows where there is oxygen. And you'll see the most growth at the top of the tube because that's full oxygen. And as you go down in the tube, you have less and less oxygen. And so you have less and less growth. And that's trying to show it less growth as you go down in the tube. In the bottom of the tube where there's no oxygen, you'll get no growth. So that's an obligate aerobe. For an obligate anaerobe, you see the reverse. In the bottom of the tube, you have the most growth, and as you go up in the tube, you get more and more oxygen. And so as you grow up in the tube, you see less and less growth. So we should have one dot right there. So there's lots of dots here, 3 dots there, and then 1 dot there. Any question about an obligate anaerobe? You see the growth in the bottom of the tube, and as you go up in the tube, there's less and less growth. Okay? For a facultative anaerobe, these organisms grow best in the presence of oxygen because they do perform aerobic respiration, but they don't require it. They can perform anaerobic respiration. They grow best in the top of the tube where it has the most growth because that's where oxygen is, and the organisms can perform aerobic respiration, and they make the most ATP from aerobic respiration. But as you go down in the tube, there's less and less oxygen, and so more and more of the cells will be performing anaerobic respiration. And in anaerobic respiration, they make less ATP, so they'll grow less the deeper down you go in the tube. And you can see there's more at the very top. It's not really showing too well, but you should see the growth sort of like a triangle. And the most growth up here, and as you go down the tube, you see less and less growth. Any question about that? How you'd see that in the tube is there'd be a thick band of growth right here, and then that band would become less and less turbid, less and less thick as you go down the tube. Any question about that? So that's the facultative anaerobes. An aerotolerant anaerobe would be an organism that doesn't use oxygen, but it has all of the enzymes it needs to detoxify the toxic oxygen molecules, like oxygen radicals. And these organisms will grow uniformly in the thioglycolate tube. There will be no difference in growth in the bottom of the tube from the top of the tube, because they can grow whether oxygen is present or whether oxygen is absent. And generally, their enzymes can handle full oxygen. Okay, any question about that? Microaerophiles are organisms that are aerophiles, so they do require oxygen. And you find their optimum growth someplace in the middle of the tube. They will be less and less growth as you go down the tube, and no growth in the bottom of the tube because they are aerophiles. And as you go up the tube, you'll see less and less growth, because although they're aerophiles, they do not like growing at 20% oxygen. Any question about them? The organisms that grow in your stomach are microaerophiles, and they tend to grow at oxygen concentrations between 2 and 10%, like Helicobacter pylori, an organism you can find in your stomach. All right, let's go back to the aerotolerant anaerobes. I mentioned they have the enzymes they need to detoxify the oxygen toxins, or the molecules which are oxygen toxins. They have things like superoxide dismutase and peroxidase to remove the oxygen toxins. Okay? Any question about any of that? All right, let's talk a little bit about culture media. In general, culture media contains the nutrients that are needed for microorganisms to grow. Medium is singular, so I have one medium, but if I have two or more different media, media is the word you use for plural. Medium can be a liquid or a solid solution, and generally the liquid solution is the same as the solid. The solid has something in it to change the liquid into a solid, such as agar or gelatin. And then agar or gelatin just makes the liquid media solid. I think you all know what gelatin is. Agar is a substance that we get from seaweed, and it just thickens things, such as the media. You can use agar in baking or in cooking to thicken what you're cooking. Generally in the microbiology lab, you'll be given medium which has been sterilized. When something is sterile, we say it contains no living microorganisms. And generally in the microbiology lab, when you get sterile stuff, it'll be because it was put in the autoclave. And usually it'll be capped in the autoclave. And an autoclave, if you don't know, is a giant pressure cooker. The inoculum are the microbes that you introduce into a medium. The culture is what we say is growing in the media once you've inoculated it. Like the microorganisms growing in a test tube, that would be the culture. There's a wide variety of media that are available. And if you were to go in the microbiology lab, I could show you a different book of all the media that they make for microorganisms. And it would be a book about that thick, maybe about that thick. And each page would be a separate media about. So it tells you how many different media there are in microbiology. Any question about any of that? Well, as I stated, the general purpose of the media is simply to provide the nutrients required for the microorganisms to grow. There has to be an energy source in the media, and that's usually carbohydrates or proteins in the media. There has to be a carbon source, and once again, that's usually from the carbohydrate or the protein in the media. There'll have to be a source of nitrogen, a source of sulfur, and a source of phosphorus. Nitrogen usually comes from the protein in the media, but there could be like nitrates added to the media. Sulfur, usually from sulfates in the media, but you can get that from protein. And then phosphorus is usually added as phosphate in the media, but you could supply phosphate like if you put DNA in the media. There might be trace elements in the media. Actually, there will be trace elements in the media. Sorry, it's almost impossible not to get trace elements in the media. I suppose if you use distilled water and you added your own chemical ingredients, you wouldn't get trace elements in there unless you were to add the trace elements. But most media, you will get trace elements from tap water, as well as if you grind up a cow, you'll get trace elements from the ground up cow parts. Any question about that? Organic growth factors may be required by organisms that are grown, like vitamin C. And if you're growing humans, for example, in the media, obviously we would need vitamin C. There are a few bacteria that do require organic growth factors. And I mentioned Neisseria gonorrhea. Strep throat, what is that? Streptococcus pylori requires organic growth factors. And we generally supply that in a specific media, like when we're growing Streptococcus pylori, we add red blood cells, and the red blood cells supply the organic growth factors. Any question about that? So if an organism needs these things, they have to be supplied by the media, and they have to be put in the media. If we were to grow a photoautotroph that fixes nitrogen from the media, meaning it can pull down nitrogen from the air and use that as its nitrogen source, what would we have to put into the media for a photoautotroph that fixes nitrogen? Come on, somebody's got to guess.
You have to put in nitrogen, wouldn't you?
No, because those organisms can fix nitrogen from the air. So nitrogen doesn't need to be in the media. Okay, but that's a good guess. At least you guessed, nobody else did. Do we need to add a carbon source to the media? Nobody studied their last lesson when we talked about the autotrophs. Because it's an autotroph, it's pulling in carbon dioxide from the air or the media, so it doesn't need a carbon source. Does it need an energy source?
Yes.
Well, yes, it does, but do we need to supply an energy source? It's a photoautotroph, so if it's exposed to light, we don't need to supply another energy source because it will use light as its energy source. So it doesn't need a light source or an energy source, doesn't need a carbon source, doesn't need a nitrogen source, doesn't need a sulfur source. Yes or no, people?
No?
Yes, you're correct. It does need a sulfur source and a phosphorus source, and it'll need trace elements. Those are all the things you need to do to supply a photoautotroph. You need to, that pulls in nitrogen from the air. You'd have to have a sulfur source, a phosphorus source. Trace elements would have to be there, but that would get probably from the tap water. And it's a photoautotroph, so you won't need to add any growth factors. Any question about any of that? All right. You guys really need to guess a little better than that. I mean, I'm not going to penalize you. And what difference does it make if how many people is in their class? Let's see, 10 other people think that you don't know your stuff if you're wrong. I mean, so what? Okay, I already talked a little bit about agar. It's what we add to a liquid media to make it solid. And you can use other things in a media to make it solid. The two most common things you can add besides agar to a media to make it solid are gelatin, and you guys are all familiar with that because it's what you find in Jell-O that makes the Jell-O solid. And the other thing would be starch. You could put starch into the media and then you have to heat it and the starch would make it a solid. I don't know if you guys are ever, yeah, you're familiar with that. When you make, what is that called?
Jello?
Jelly? No, not jelly. That you put gelatin in jelly. When you make gravy, you add starch or flour to it to thicken the gravy. And if you were to add a whole bunch of flour or starch to the gravy, it would make it solid, okay? But when we're adding the starch to the gravy, it thickens the gravy. And that's why it's not the thin juice of the meat. We call it a gravy when it's not really a solid, but it's not a liquid either. It's a semi-solid. So it's thicker. So those are the two things that, well, besides agar, that we use to thicken things in microbiology. And agar is almost always used. Gelatin is a very specific test we use it for, and the same thing with starch. It's really only used in a specific media. Usually the agar is not digested by the bacteria, and that's why we use agar. Dr. Koch initially invented solid media, and the first thing he used was actually a cutting from a potato. And I don't know if that was a raw potato or a cooked potato, but he cut the potato and then he grew his microorganisms on it. And as you can imagine, it wasn't very good. Yeah. And he then started using Jello, where he put Jello into his media. And the trouble is that there are microorganisms that can digest the Jello, and then he got a liquid mess because the media was no longer solid. And he didn't like that too much. And it was the wife of one of his assistants. I can't remember if that was Petrie or somebody else, but his wife suggested, why don't you try agar? And agar is a substance that bakers or people who cook add to thicken things. It's not used very much today, but it still is used to do that. And it was used more in the past. And agar worked very well because microorganisms don't digest it. And that way the media stayed solid. And all it is a substance we obtain from seaweed, okay? And for some reason, microorganisms haven't learned to digest it yet. And so we use it to solidify our liquid media. You usually find it in a tube. Let me see if I can bring up something so I can show you. There's two types of tubes. There's a deep. You know, this won't be the best because I don't think I can draw in this. Let me see. There is a way to draw in here, but I'm not sure how to do it. So let me see if I can bring up something else. I need a program for drawing. Just a minute.
Use paint.
Yeah, I know, but I gotta get something to open something so it will open up Paint. There we go. Let's get rid of that. Come on. So, well, that'll be good enough. So you can have a tube, and it's a deep where we have the media, if it's solid, it goes across like that. You can also have a tube where we call it a slant. That's trying to be a tube there. And in a slant, we grow the media. Oops, that's not a very good slant. Something like that. Or you could grow it like that. Let me get the eraser. Oh, that's not what I wanted to do. Darn it. Let me see if I can correct this. There we go. Not a very good slant, but where the slant is a complete slant, and that's the entire media right here. Or you can get it where it's partially a deep, and then you have a slant on top of it. So these are called slants. And then the other tube, where the media is going straight across, that's a deep. And I've got that listed here. Their tubes are usually in slants or deeps. I don't want to talk about what temperature agar solidifies or does anything on. There's different types of media. Are there any questions about agar or the tubes? There's different types of media. As I mentioned, we've got a book about that thick that has different types of media. There are chemically defined media. Let's blow that up so you can see that. And in a chemically defined media, we know the exact chemical composition of what's in the media. So everything in the media we add, like this is a very simple media based on glucose. and it has ammonium phosphate added, sodium chloride, magnesium sulfate, potassium phosphate, and then water. And if we use distilled water, we're not getting trace elements. And those are the only molecules in this media. Now here's a much more complex media, but it's still chemically defined. And we're adding amino acids, organic growth factors, sodium thioglycolate for a reducing agent, and then water. And it has a large number of salts to add things to it. And a media that requires many growth factors would be used by a fastidious organism. That's one that you need to add many growth factors in order for that organism to grow. Like I mentioned, the Streptococcus pylori is something that we add red blood cells to the media to get it to grow, because Streptococcus pylori is fastidious and it would not grow in a normal media, which, you know, you grind up your cow and then you make a beef extract and you add the water. A Streptococcus pylori won't grow in that. E. coli would, but Streptococcus pylori would not, because it's fastidious, and it requires a lot of growth factors. And what you can do is add red blood cells, and then whatever the growth factors are required by Streptococcus pylori, the red blood cells supply the growth factors. Okay, so chemically defined media are not used very often in microbiology unless we're growing autotrophs, because an autotroph generally can make all of the molecules they need from sugar, and of course they can make their own sugar. Okay. Most of the other organisms we grow in microbiology, we have a complex media. And these media contain extracts or digests of meats or plant protein, and they're providing the nutrients, the vitamins, and the organic growth factors that are required for those organisms to live. A very simple one is shown here. Let me get that out of the way. And it's nutrient agar. It has a peptone source. That's a partially digested protein, and that probably comes from soybeans. And then ground up cow, meaning we call it beef extract, and it's supplying a lot of the nutrients as well as the carbon source and the energy source. And the peptones are supplying the carbon source and the energy source as well. It has sodium chloride in it to help the osmotic pressure, and then it's supplying sodium and chloride. And then agar to thicken it, and then water. And if this is tap water, which is probably what it is supplying trace elements like minute amounts of copper and iron are in the water. And the minute amounts of the trace elements would also be in the beef extract. The point is, we have no idea what molecules are in this. For example, I don't know, is chlorophyll in the beef extract? It's possible, because the cow was eating grass. So there might be chlorophyll in the cow, and then we grind up the cow. That might be supplied. My point is, different cows would have different molecules, like a cow growing in Arizona compared to a cow growing in Colorado, because they're eating different things, and different molecules would be in them. Even different batches from the same company would have different molecules in them. And we don't know what all of the molecules are in a complex media, but then again, we don't care, because all we're trying to do is add beef extract and peptone so that many organisms would grow. Now in the lab, we do supply you occasionally with nutrient agar if you were to go into the lab, but most of the time we would supply you with nutrient agar with yeast extract, and we call that NA media, nutrient agar, N, A, and then Y for yeast, and E for extract for NA. And it's a little richer than this, and you're getting nutrients and organic growth factors from the yeast extract. Any question about that? So the carbon source and the nitrogen source is largely coming from the beef extract, but it's also coming from the peptone. The sulfur extract is coming from mainly the beef extract. The trace elements, of course, coming from the water and the beef extract, a little bit from the peptone. And these are the media we use for growing heterotrophs, like E. coli and fungi. Okay? And they have an organic carbon source, like the beef extract and the peptone, for the organisms to use. There's different types of complex media. Most of the media you use would be something that would be an aerobic media, meaning that it's intended to grow in the air, and so the microorganisms would be exposed to oxygen from the air. If you didn't want that, you were trying to grow anaerobes, you would have an anaerobic media, and that would be important for growing obligate anaerobes, organisms which must avoid all exposure to oxygen. And you grow those organisms in a reducing media. A reducing media is just a media containing a chemical to remove oxygen. Sodium thioglycolate is the only one that we've discussed, and probably the only reducing media we'll ever discuss. And sodium thioglycolate combines with oxygen in the media and then removes it. The result is that at the bottom of the sodium thioglycolate tube, there will be no oxygen. Any questions about that? When you're autoclaving that media, that tends to make the oxygen leave. And so when you first get the tube out of the autoclave, there would be no oxygen in it, but oxygen from the air will go into the tube, and that'll change the thioglycolate tube. And that's why it has total oxygen around 20% at the top of the tube. And then as you go down the tube, you'll get less and less oxygen. And at the bottom of the tube, you'll have no oxygen. Any question about that? There are different types of media. Many of the media that we've talked about so far have been general purpose media, like the complex media, nutrient agar, or nutrient agar with yeast extract. These are general purpose media. General purpose means is that we use it generally to grow many organisms. Doesn't grow everything. Actually, there's no media that grows everything. But like Streptococcus pylori will not grow in nutrient agar or nutrient agar with yeast extract because it's fastidious, and we have to have a very complex media adding the growth factors that it requires. But the point is, general purpose media are non-selective, and we can grow many things in them, and they're also cheap to make compared to a fastidious media or even an anaerobic media. And generally they're cheaper than a selective media. A selective media is a media that has some ingredient, some chemical that selects for certain things to grow in it and selects against other things from growing in it. Okay? So it halts or inhibits the growth of a particular group of microorganisms and does not affect the growth of other microorganisms. An example of a selective media is one that has 7% sodium chloride in it. 7% sodium chloride selects for organisms that can tolerate or grow in 7% sodium chloride, and then inhibits organisms that cannot grow in 7% sodium chloride. Most microorganisms cannot grow in 7% sodium chloride because the most they can grow in is like 1 or 2% sodium chloride. But halophiles, organisms that like sodium chloride, halo, salt, and then phile, lover, so a halophile is a salt lover, can grow on a 7% sodium chloride plate. So that's a selective media. And if you don't know, the microorganisms growing on your skin can grow on a 7% sodium chloride plate because the microorganisms growing on your skin are halophiles. And why is that? Why is the microorganisms on your skin halophiles?
Because we sweat a lot.
Yeah, we set and release sodium chloride when we sweat. And so your skin can get up to 20% sodium chloride, depending on the last time you showered and how much you've been sweating, okay? Any question about that? Okay, there's selective media where you can, well, let me state this. Selective media where you can grow certain organisms on it and then other organisms don't grow or at least grow poorly. There's differential media, and this allows us to see differences between different species. So it has a chemical in it, or more than one, that reacts differently to different species, and then that chemical shows up differently on the plate. Does that make sense? One plate that's a differential media, and it's also a rich media, is a blood agar. With blood agar, it's differential because if you're growing something on blood agar, you can look for its hemolytic activity. And there's 3 types of hemolysis, or 3 types of hemolytic activity. There's gamma hemolysis, and that's supposed to look like a gamma in the Greek alphabet. And gamma hemolysis is no hemolysis. Let me blow that up so you see it even better. And the blood stays in the plate all the way to where the organism is growing. So there's no hemolytic activity. In alpha hemolysis, you can see darkening around the white growth. So this ring of dark here, and then inside the alpha, it's dark. That is where there's partial lysis of the red blood cells. So it's partial, and the plate looks darker where there's partial hemolysis. Sometimes it looks greenish in color where there's partial hemolysis. But there's some blood cells not being lysed, and so you can't see through the partial hemolysis. And that is alpha hemolysis. Okay? And then there's beta hemolysis, and the white here is where the growth is. And then this ring is very light, is where all of the red blood cells have been lysed. And so we can see through the media because the red blood cells have been lysed, and so light can get through. Okay? Any questions about beta hemolysis, alpha hemolysis, or gamma hemolysis? Now you'll notice that this is the growth, and then this clearing here is the beta hemolysis. You'll notice we have a dark ring around the beta hemolysis. What that is alpha hemolysis, and there's usually a ring of alpha hemolysis around the beta hemolysis, meaning this is partial hemolysis right there. And that's something normal, but we call that beta hemolysis. And that allows you to determine the differences between 3 different species, like, oh gosh, I'm trying to think of a species that's, E. coli is a gamma hemolysis. It does not normally lyse red blood cells. Streptococcus epidermidis is alpha hemolysis, although Streptococcus epidermidis is very weak on giving alpha hemolysis. Sometimes that'll look like gamma hemolysis, but I can't think of another example that is alpha hemolysis, so that's why I'm going to use it. And if it's done well and correctly, you should see a little bit of alpha hemolysis with Streptococcus epidermidis. And then Staphylococcus, did I say Staphylococcus epidermidis? Staphylococcus aureus is a beta hemolytic bacteria, and so you'll see beta hemolysis with Staphylococcus aureus. And that's how a differential media can allow you to see differences between 3 different bacteria strains. Usually on differential media, you'll only get 2 differences. Like one will be dark and the other won't be a change, and so it'll be light. Now I said there's selective media and there's differential media where you can have them together in the same plate, and that we call selective and differential media, where it's both selective and differential. Eosine methylene blue is a selective and differential media. It's selective against gram-positive bacteria because gram-positive bacteria don't like growing in the dyes eosine and methylene blue, and these 2 dyes are put into the media and they get into the gram-positive cells and they prevent the gram-positive cells from growing. And that's why it's selective. Any question about that? It's differential. Sorry?
So only gram-negative bacteria would grow in that?
Essentially, gram-negative bacteria would grow in this plate. Now, it's not perfect on its selection. So sometimes the gram-positive will be inhibited, but not inhibited 100%. So you would see slight growth on a certain bacteria that's gram-positive. And then the gram-negatives usually are not inhibited, but sometimes they will be inhibited, okay? But generally on an eosine methylene blue, the gram-negative would grow and the gram-positive would not. And that's only the way we're going to talk about it. But the selection is not 100%, okay? Eosine methylene blue is also differential, and it's differential because it has a pH indicator in it and the sugar lactose. And so it's differential on whether it's a lactose fermenter. The bacteria turn a dark color with eosine methylene blue plates if they ferment the sugar lactose. And what's happening is that when the sugar lactose is fermented, it produces acidic products, and there's a pH indicator in this media that turns dark when the pH is acidic. Is that clear? So the cells are dark colored, that's shown right there, when the colonies are dark, when it ferments the sugar lactose. And that will only happen in a gram negative, because generally speaking, the gram positives aren't going to grow on this plate. All right, so you can't say if you put a gram positive on this plate and it doesn't grow, you can't say if it's differential for lactose fermentation because the gram positive isn't going to grow on the plate. Is that clear? All right, but if it's gram negative and it doesn't ferment the sugar lactose, it's differential because instead of having a dark colony, the colonies will be normal colored, and this colony is shown best in this picture here, because that you're getting a shadow there. So that's white and dark. But this one is all white, and that's because it's not fermenting the sugar lactose, and so it's not becoming dark. Any questions about why eosine methylene blue is both selective and differential? All right. Yeah, let me go ahead and finish this one last topic and then we'll stop. Another type of a media is an enrichment media, and this encourages the growth of certain things to grow and discourages the growth of other things from growing. So an enrichment media is a selective media, but we use it not really for, well, selection. We use it for enrichment, meaning that if we're wanting to see a certain thing, then we grow it in an enriched media that helps select for what we're trying to find. Any question about that? So an enrichment media increases the number of desired microorganisms, generally to a detectable level. So if you're looking for something causing a patient to be sick, like they have gastrointestinal distress and you're looking for that, the normal human microbiota will be so many different species, and then the pathogen causing the illness could be so low that if we were to just plate it out and then streak for isolated colonies, you might not ever find the pathogen. So you put it in an enrichment media, and then that will select for the pathogen. And that's particularly useful if the targeted microorganism is only a small proportion of the total population of cells. And generally, when we're looking at a human pathogen, at least in the mouth or the gastrointestinal system, the pathogen could be at low numbers, and then to find it would be very hard. The same thing if you're looking for a soil bacteria. It'll probably be in the soil at very low numbers compared to most other organisms. And so you need to grow it in an enrichment media. For example, if we were trying to get a phenol degrading bacteria from the soil, you would probably find it, but it would be in the soil at a very low number. And so streaking it out for colony isolation, you'd never find it. What you could do, if you're looking for a phenol degrading bacteria, is grow it in a media where the only carbon source is phenol. And then that would enrich for bacteria that are phenol degrading in the soil. And what you do is, let's see if I still have that open, is you have a media and you put a little bit of soil in, and then you grow that in the enriched media that the only carbon source is phenol. And then after that grows up, you get another tube of it, and you take some of this and put it into the second tube, and then you further enrich it. So let's say you're looking for something in the soil that is 1 in 10 to the 5th. Well, to try and find this from a colony streak would be impossible, because the best you could do would be maybe 1 in 100. So we have to enrich it. And if this enriches it 10-fold here, we've changed it to 1 in 10 to the 4th. And we put it in this here, now this is 1 in 10 to the 3rd. So I should say 1 over 10 to the 3rd. Still not good enough, but we could do it again. So I'm going to go like that and put it here. Now, after growing it in enrichment media 3 times, we've now got it up to 1 in 10 to the 2. We can now plate it out, and we have a chance of finding our bacteria. The chance of finding it isn't very good. You'd have to look at 100 colonies before you'd find it, but at least you have a chance of finding it, okay? If we were to do it one more time, let's put another plate here, and if it were to grow 10 times, meaning enriched 10 times, here we got 1 in 10, and that would be an excellent chance of streaking it out and finding it. We'd only have to look at 10 different colonies, and then we would get the phenol degrading bacteria of interest. Anyways, that's how you use an enrichment media. Any questions about that? Okay, well, if there's no questions, I think I'm going to end it here, and this went past a little bit. I'm going to be a little bit late. Let's make it 2:35 for the lab, because I need at least 15 minutes to get this to download. All right, if there's no questions, I'm going to end this.